Okay, go.
Keep going?
Yeah.
Okay. Okay, so our presentation is about more experience equals bigger brain. And this is a study on rats, but relating to humans. And I'm Kristen,
Taylor.
and we have Dane and Jordan here. Okay, so the study that we're looking at was published in 1972 by Rosenzweig, Bennett, and Diamond. And the overall question that they were looking to answer was, does experience result in observable changes in the brain? To answer this question, they exposed rats to different environments with various degrees of stimuli, and Taylor will talk about this soon. And so the researchers' question was important for several reasons. For one reason was that it tested the importance of a stimulating versus a non-stimulating environment on cognitive development. And so this could give insight into what is the best kind of environment to raise a child in. Further, it was a unique study in that it connected cognitive development to actual physical changes in the brain structure. So this allowed the study to gain insight into how learning and memory, which could be considered subjective processes, relate to concrete or objective changes in the brain. Next slide. Okay, so this study relates to several concepts that we've looked at in class already. So first, it relates to the nature versus nurture debate, and that is about how genetic versus environmental factors impact human development. And so as we have learned, a lot of researchers have moved away from the either-or approach to this method and see both nature and nurture as important factors in development. And so this study had the potential to support the nurture side of things, since it looked at the brain changes in response to exposure to a certain environment. In class, we also talked about plasticity, which is the ability of the brain to change in response to experience. So as you can see from our title, this directs exactly to our study. So if you remember, the synapses are the spaces between nerve cells that allow for conduction of a signal to take place. And so this process of eliminating unused synapses is called pruning. And so this study was just important in learning about this and seeing how the synapses can change in response to experience. Another topic we looked at was parental versus non-parental care. We learned that in a daycare setting, kids tend to do well on future tests if the setting has more stimuli than they would normally get at home. And so this study looked at the effects of stimulating environments, which relates to this idea overall. And so the researcher expectations was that they were just looking to see physical differences in the brain due to different environment settings.
Methods. This was an experimental study. They used an experimental study because it showed how environmental factors, how they affect what their effects on the brain were. In this study, they used laboratory rats. There are lots of different reasons why they use laboratory rats. First of all, for convenience, they're small and inexpensive. Also, because they have large litters. So when they did the experiment, they were able to take 3 rats from each litter and put them in the different environments, so that way they had the same genetic basis. Also, the brain is smooth compared to more complex animals. The brain is folded, whereas in rats, it's smoother and easier to dissect. And also, it's more ethical to be working on rats than it is on humans. Procedure. This experiment was redone multiple times. They also used different strains of the laboratory rats and also on gerbils. They took 3 rats from a litter, males, 25 to 105 days old, and placed them in the 3 different environments. They had a standard lab environment, which is here. It had several rats with adequate food and space for them. The enriched environment was a larger cage, which is here, and it had more rats in it, and also they had playthings, which were randomly chosen out of a group of 25, and they were switched each day. And then the impoverished environment which is here, where a single rat was placed in a smaller cage and kept in a separate room, isolated. After a predetermined set of time, which was usually somewhere from 4 to 10 weeks, the rats were then dissected. To eliminate dissector bias, the person who was dissecting the brains didn't know which cage the rats came from. They were only able to identify them by their code number.
Does that mean anything to you?
Okay, what did the researchers find? The research resulted in the enriched environment rats had greater weight of their cerebral cortex, and this includes a greater ratio of cerebral weight to their overall brain weight. So this figure right here shows in all 16 studies showed a greater percentage difference of cerebral weight to overall brain weight. So this increased in the enriched environment, and in this greater weight of cerebral cortex, they found the amount of DNA per milligram in the tissue decreased while the RNA stayed the same, and this suggested higher metabolic activity. So there was more neural activity going on in this enriched environment rats. They also found greater thickness in the cortex and greater activity of acetylcholine esterase, which is a neurotransmitting enzyme that basically recycles a neurotransmitter. So the neurons could conduct faster nerve pulses. They also found that the occipital cortex of the enriched environment rats were heavier. And finally, they found that they had larger synapses as well, which basically there was more area of the synaptic cleft for the signal to be transducted. And then, so this all concluded that the researchers' original prediction that the enriched environment would cause a change in brain development was met. And further research by Bennett in 1976 showed that brains of impoverished creatures could be improved if moved to an enriching environment. So, And other research showed by Hobson in 1984 showed more stimulation caused an increase in neural tissue. So people who use their brains don't lose them. So even as you get older, you can still use your brain and you're not going to lose that activity. And finally, they found that brain development balances not only like brain chemicals, but also an individual's life context, so their genetic tendencies, and the balance of an enriched or impoverished environment.
All right, so why is this study important, or why is it a landmark study? It showed that intelligence and memory are related to nurture, but I think we need to remember that nature is also important. The study is very focused on nurture and the effects of nurture, but we can't forget the effects of nature, because if there were no effects by nature, every single rat would have the same brain weight. Next slide. So instead of this, where nature and nurture are competing, we can see my amazing picture editing skills and flipping it, and we can see that they should be holding hands and working together. And then the second reason why it's a landmark study is the implication for memory. And providing a stimulating environment can help with preventing or slowing memory loss, so it could be used in situations with older people that have problems with memory loss. We could help provide them a stimulating environment, and it could possibly help them. And then also it showed the effects of brain plasticity, and it showed that the brain is able to change in response to experience. The brain is not something that's just there and it doesn't change at all. So what were some of the limitations of the study? There were some possibilities of other variables having effects, such as stress or the handling of the rats, but both of these were later controlled for in separate studies, and they were shown to be not factors in the brain size. And also, it's an artificial environment, and the lab environment is very different from a natural environment, and it's hard to look at a natural environment, especially for rats or mice, because we can't just take them and try to determine whether or not they had an enriched environment. And then another problem was the use of mice in a study, and it's hard to generalize it to humans. We don't have people mice, like this picture. That'd be kind of creepy. So it's kind of hard to generalize this study for rats and put it to humans. And then it's also unethical to carry out this study on people. We cannot randomly assign people to a impoverished environment. And then also, what age does this apply to? Is the enriched environment only important while the brain is developing, or is it important for the entire lifespan? And so some recommendations we have for the class. The first, and probably the most important, is when raising children. I'm assuming most people in the class will have children at some point in their life. And make sure you provide them with an enriched environment and don't let them do the same thing. And just provide them with a lot of different things, like read to them, give them toys, like just let them experience new things. I think one of the most important things for the rats in the enriched environment was that they gave them new toys every so often. They didn't just give them the same things over and over and over. And our second recommendation is stimulate your own brain. Don't be a couch potato. Go out and do something, experience something. Go play, go read, go write. Don't just sit on the couch and watch TV all day. Go do different things. And the third is, especially with the further research, is in adopting children. Some people would probably be worried in adopting children that they come from a bad background and they wouldn't be able to catch up or anything, but the further research did show that the mice that were later placed in the enriched environment, when they started off in the impoverished one, were able to catch up. Are there any questions? No? Thank you.